Welcome to the NCBI Now workshop, Measuring Transcripts with RNAseq. My name is Jonathan Pevsner. I'm really excited to present this to you. Our topic is gene expression studies using RNAseq. Here's the table of contents. We'll talk about the background and motivation, how to organize your directories, then we'll introduce an RNAseq workflow. We'll download, compile, and install a couple software tools, get some data of interest from NCBI's website, as well as a reference genome. We'll do an alignment of the RNAseq reads to the reference, and then we'll go ahead and view it. We'll end up by taking a look at which genes and which isoforms have been expressed using this technique. So here's the background. When I think about RNA, I think about gene expression. By the year 2000, microarrays had emerged as a major technique to measure RNA transcript levels in samples such as tissues or cell lines. A decade later, by the year 2010, RNAseq had emerged as a new method based on next-generation sequencing technology. It offers many advantages. It allows you to assess any RNA species in a sample, rather than focusing on particular transcripts that have been pre-selected to be represented on the surface of a microarray. The dynamic range of RNAseq is really excellent. You can look at transcripts that are expressed at very low levels, ranging all the way to high levels. Most protein-coding genes, not to mention non-coding genes, have multiple exons that are alternatively spliced. RNAseq lets you see these. You can measure their levels of the whole gene and/or of isoforms. Also, when you annotate the exons in any genome, including the human genome, it's very challenging. By annotating the exons, I mean determining what the structure of genes is. And RNAseq allows you to identify novel splice isoforms you might not have seen before. These might not even be annotated in the genome. RNA itself is expressed in a context-dependent manner. What this means is that your genomic DNA is usually thought of as being similar across the somatic cells in your body. For example, the genomic DNA in your brain is very similar to the DNA in your liver or your toenail. RNA, however, is always expressed at some time and place. For example, mouse brain or mouse liver, human brain or human kidney, early in development, late in development. It may be in the roots of a plant that you're looking, or maybe it's in cell lines plus and minus treatment with a drug. So we usually think of RNA expression in terms of comparison of two or more conditions. And this is shown in the picture, where DNA is transcribed into RNA. It may be translated into protein. The RNA may be made more stable in the form of complementary DNA or cDNA, and it may be then studied on a DNA microarray or by looking with RNAseq. Let's get a better feel for how RNA is expressed across the body. We'll take an example of DNMT3B. It's a gene on chromosome 20. You enter that name or that symbol as a query on NCBI's website and set the database to gene. Here's the direct link. You can scroll down a bit to see a browser, and on that browser you can see these bars here in green that represent exons. And furthermore, you can see the arrows showing you the direction of transcription from left to right, or 5' to 3'. These various transcripts have accession numbers on them. Up to the right, you can see a gear-shaped icon labeled tracks, and if you go configure tracks, you can then get access to a menu that includes expression on the left side, where we can find literally hundreds of RNAseq experiments available to you. All you have to do is check the ones that you're interested in, and then configure. You come back then to your browser, and you've added these many tracks. And this tells you information. For example, you can see some of these tracks are populated with very high coverage, where others, such as adult cerebral cortex, has low levels of expression. Here at the bottom, we can see adult human skin having high levels of expression. You may also notice that some of the exons may be differentially expressed in a tissue-specific manner. So you can see some signal off here to the left that you don't see in many of the other samples. I'll do this very briefly going live from the homepage of NCBI. We can go ahead and paste in our symbol, search, go to the gene database. Here we can select human. This is sorted by relevance. And on this page, we're in NCBI gene as shown in the top left. You can see that browser as we scroll down just a little bit. Here's the gear icon. We can go ahead to tracks, configure, And then on the expression side here, we can find several hundred different RNA-seq exon coverage or even intron coverage experiments and choose the one you're interested in. And this is a really great way to learn more about your favorite gene of interest and how it's expressed. Okay, so our goals in this workshop, we're introducing RNA-seq, finding samples, finding a reference genome, doing alignment, and then seeing what is regulated. Our next topic is how to organize the directories, and this is pretty straightforward. We'll continue to use Mac OS X. This is my MacBook Air that I'm working on. You may also do this on a Linux machine, and if you're working on Windows, I presented several different options in the earlier presentation on DNA sequencing. So let's make a main directory for our RNA-seq work. And as a reminder, when we have the tilde character, that refers to the home directory, which in my case is /users/Pevsner. And so we can do mkdir to make a directory called, in my case, projects/NCBI now RNAseq. You can give it any name that you'd like, and then make a few more directories for projects, make directory for NCBI now, and then go ahead and do one for raw data, reference, analysis. I made one for HiSat as well right in here. And then for programs, I typically put programs into their own directories in one area. And so here, just to review, I can go ahead and cd home. I can do pwd, which means I'm at users Pevsner. And then I can go to projects, NCBI now, RNAseq. And in there, I can go pwd, confirms where I am, ls, shows me what I have there, ls-lh, shows me that these are all directories as indicated by the letter D on the left side. And if I wanted to make a new directory, I could make a directory called test. Then now if I do ls-lh, using the up arrow to recall recent commands, I've made one called test. And so that's easy to do. Let's return to the HTML, and I'd like to introduce our RNA-seq workflow. We can think about workflows in general as follows. Here's the upper part of one. We begin with a biological question, experimental design, and we go ahead and isolate RNA. These are really important stages, and it's a great idea to collaborate with a statistician and a bioinformatician if you're a biologist. One question is, what's the appropriate sample size? And that answer may depend on the magnitude of the effect you plan to look at. But please avoid doing just one sample in a condition. For example, many people think that 3 is a reasonable sample size for each group. Others suggest 5, 10, or even more samples. Of course, cost may be limiting. Another thing to remember is, if you have control and experimental samples, be sure not to process the RNA for the controls on a Monday and the experimental on a Tuesday. Why is that a problem? It's called a perfect confound. It means that any differences you see could be due to date, rather than the comparison of control and experimental. In the next part of the workflow, we can make a library on the left side, do next generation sequencing, and the topic of our workshop today is to map reads and quantitate expression levels. All this is the RNA-seq side. I'd like to point out how parallel it is to the microarray side, where we label, hybridize, wash, scan, do image analysis, and normalize, and come up with the same kind of answer, what happened to the RNA transcripts in my experiment. Next, we do hypothesis testing. That is to say, what are the p-values or probability values we ask, for example, for a series of 20 or 30,000 transcripts for each one? Is the expression level in the control group and the experimental group the same? This is the null hypothesis. Can we reject the null hypothesis at some significance threshold, such as 0.05? And so this is hypothesis testing. In the middle is exploratory statistics, such as multidimensional scaling, principal components analysis, clustering. These are different ways to explore your data. And on the right is classification, where you can ask, does this expression profile correspond to that seen in individuals with a disease phenotype, for example? I would suggest that biological confirmation is always important, for example, quantitative real-time PCR, to confirm your findings. And then we hope at the end to come to biological insight, perhaps better diagnostics and therapeutics, or strategies for therapeutic intervention at least. Now here's another slightly more detailed RNA-seq workflow. We can think about dozens and dozens of software packages. On the left, experimental design, RNA acquisition, and data acquisition. We're going to get millions of short reads. We'll map them, and there are many tools available. The one that we've picked for today, HiSat, is really robust, and also it's extremely fast. We'll get data including mapped and unmapped reads with respect to some reference genome, and we'll go pull a reference genome as well. The next steps include summarization and normalization. We're going to try to find out what happened across different exons or across different genes, what happens in terms of junctions between different exons, and then we'll get a table of counts that we can study, and it will be important to normalize the data. For example, if a gene is 10 times longer than another, there may be 10 times more reads, not because it was more highly expressed, but because more reads can map there. So we'll want to normalize things. And then we come to differential expression testing, and again, there are many software packages here. We'll get a list of differentially expressed genes or transcripts. We'll try to interpret this, and beyond the scope of the workshop, we can test for enrichment of particular functional groups and study networks. In the end, again, I recommend validation. What's the significance of this? Well, we're using HiSat, which is one of the dozens of software tools available to do alignment. It's extremely fast. I recommend reading the paper on HiSat, which describes that it uses two different kinds of indices. They're called FM indexes, a global index and then an additional 48,000 small indexes, which help contribute to the great speed of this program. So sometimes RNA-seq work takes maybe 10 hours to study a sample. Here that may take 10 minutes. I don't want to exaggerate that difference, but it's much faster. And all these programs differ in their speed and accuracy. I would also like to mention a methodological issue. You have here, and I've made this image of genomic DNA, which has exons with intervening introns. At the genomic DNA level, we could take our RNA-seq reads and align to the whole genome and find where the mapping to exons occurs. This might even show us new splice isoforms if we have reads mapping to a region that hadn't been previously characterized as an exon. That's great. In general, the genomic DNA may be transcribed to messenger RNA or transcribed to RNA which is processed further. And so here you have splicing together of the different exons. Another strategy with RNAseq is to take a file not of the whole human genome reference, but instead a reference of the transcriptome. This is relatively more straightforward to do. Our next task is to download, compile, and install various software tools. You can get HiSat. The instructions are given here. The key publication, note to go to the website for HiSat and download it. You can put it into your path. You can also download ngslib. It's a zip file obtainable from the GitHub website, which will facilitate your use of HiSat to download data from NCBI directly. And this is a really nice thing to do. The websites involved here do a great job of explaining how to download this software. And also here I've written out some of the commands that I used as I downloaded it, including making a directory for HiSat, going into that directory, moving my downloaded files, for example, NGS master, the dot means move it to here. Go into that directory and do a configure command. Sudo is for a super user, and that is giving you permission to do this configure. And then you can use a command that's given in the documentation for NGS master, and HiSat also gives various documentation. The other program I'd like you to install is Stringtie, and here the key website is given again, the key publication with its PubMed ID, and you can download the pre-built Apple OS X binary, and that's just a straightforward thing to do so that you then can take the file, unpack it, and you can put it into your path and you'll be able to run it. Let's find some RNA-seq data. We'll use reads from the very well characterized cell line GM12878. The corresponding genomic DNA from that individual is NA12878. So let's enter the query, GM12878, at the homepage of NCBI, take a look at the records that are shown up in the Entree database, and you'll see a link to SRA with 1614 entries. Go there, and on the left sidebar, restrict the results to RNA. Now you can go use the Run selector under the Send To menu. The Run selector is great. You click Go, and it shows you that there are several hundred runs. By the way, if you start with NA12878 instead of GM12878, there are far fewer runs available, so we want those cell lines with the GM number. So the main thing you need to know, and the main thing you need to do, is to find the SRR run accession number. There's a whole list of them here shown in the run selector. Those are the ones we're going to use either with fastqdump or by running HiSat. In the alignment below, we'll use SRR764785. So when you choose one of these, we don't really mind which one we take. You can select the run and click for more information, or the biosample, or the bioproject. You can learn more. We'll use several other ones as well. On the left side, there are these great facets, and they let you restrict your search. For example, if we check megabytes, we can make sure the file isn't too big or too small. So go ahead, and I've even tried getting a few gigantic files just to take a look. And still, the main idea is we don't want to bring huge data sets down to our computer unless we need to. We're going to use fastqdump to download sequence reads in the fastq format. And so I'll go do this here. Fastq-dump is the SRA toolkit utility. And if you have paired in reads, then you need the split files argument. And it tells you that it reads those, it writes those, and you can check the size of what's downloaded. So here, just to do it live, on the command line, we can go fastqdump split files and enter, and then this will be downloaded automatically, and you'll have it on your computer. So you pick the SRR number of interest. You can compress your fastq files using gzip, and so the command is simply gzip and then the name of the file. What that does is can turn a file from 7 gigabytes down to just about 2 gigabytes, and that's great. HiSat is capable of handling either uncompressed or compressed reads. If you do download paired-in reads, I'd like to suggest that you use the word count utility to confirm that the two files have the same number of lines as they should. That's shown here, for example, where there are 188 million reads in the fastq forward and reverse. When we say forward and reverse, we mean that a piece of RNA has been turned into cDNA and can be sequenced from one end or in paired-end sequencing from both ends. And that can be very helpful, for example, to see if they map to an appropriate location. If they map to very distant places, those are discordant reads, and we might be interested in seeing those. But single-end reads are very useful too. So here's an example of a typical fastq file using the head utility. And by the way, as I'm telling you about this, you might notice that on the prompt, nothing's shown up yet. The cursor hasn't returned because this dump is continuing, and that might take a little while. So when you look at a fastq file, you can do head and the file, and you can see the typical format of a fastq file. And by the way, while you're waiting, I'm going to open up a new window. And on that window, I'll make it a little bit bigger. I'll type top, and that tells me about the processes that are running on my computer. Here's fastqdump taking up 96% of one unit, one CPU. And so this is a way that you can just tell that your computer is actually at work and it's doing what you think that it's doing. And indeed, because I've already downloaded this, I'm going to cancel it right now. So here's an optional dump. I don't really think you should do this unless your computer has lots of space. I took a huge file and brought it down, and it turned out that it was so big it was 35 gigabytes for the forward and separately 35 more gigabytes for the reverse read. So it's just an enormous file, and that's what we really would like to try to avoid. Okay, next we're going to obtain a reference genome and index it. There are lots of places to find and download reference sequences. NCBI, Ensemble, and UCSC are three of the leading places. You can also get corresponding annotation files. You can download a whole genome sequence or individual chromosomes. So, for chromosome 20, we're going to do this from Ensemble. And if you go to the home page of Ensemble, click Downloads, and go to Databases, you can very easily get to this page, which shows you for different species, such as human, DNA in the FASTA format, which is what we want, or gene sets, and these are useful as well. Follow the link, and you can get FASTA files for individual chromosomes. You can also get a GFF3 file that we'll use at the end as we're doing our annotation of our findings. So, download Ensemble Chromosome 20 reference file in GFF. The names of them are given here. Homo sapiens GRCH38 is our build. That's a GFF3, and here's chromosome 20.fa for FASTA. For those who are interested in using NCBI, it's also great to go there. I recommend that you go to the home page, go to Assembly, find the human genome by entering that in the search box, and go to the current build, GRCH38. The bottom of this page shows you a list of FASTA formatted files for each chromosome, and then you also have on the right side GenBank FTP site and by going there, you can also download your files, such as the whole genome, and you can also find whole chromosome. So once we've got our FASTA file, we want to use HiSat build to prepare it. You can see the usage and options just by copying that command. And that tells you how to do it. It says no input sequence was specified, but it's saying that we want to have a reference file and that here are some of the many options that you can follow. So when we actually use it, we do HiSat-build and we do the name of the chromosome 20 FASTA file. We specify an output, and all that I've done is add _HiSat to the end of that. This takes less than a minute for a really small file, but it can take many hours to build with a large file. What's the output? Well, you can see a whole series of files, and we don't need to know a lot about what these are. When I did this build with my gigantic FASTQ dataset, it took many, many hours to do the same thing. So at this point, we've gotten our reference chromosome, it's convenient to use the small chromosome 20, and we're ready to do our alignment. We are ready to use HiSat to align reads. If we type HiSat, we can see the usage and the various options. I'll go ahead and do that. And you can go explore these to see more information about how the program works. The main command we'll use is here. HiSat-X refers to the particular reference sequence. --SRA accession. We're going to specify an SRR1 accession and then an output file. And so let's take a look at this. We'll go copy, paste, and run that. And I will give this an extra character. So what we have here is a command in which the X instructs the aligner where to look for the reference file. ... means go up a directory, then back down into reference, and we have Homo sapiens GRCH38, chromosome 20 FASTA, that's been prepared by HiSat with the HiSat build command. It's a long name, but on the other hand, that's descriptive, so we know what we've got here. The next thing is the SRA accession number, and this is really great because once you've decided which runs you want to look at, you don't need to download them. You can just run them here right through HiSat. -S specifies the name of the output, and again, this is a long name, but you can make it informative so that you know what it is you're aligning. This was a HiSat project. I put in the particular accession number of the run that we're doing, and then the genome build so that you're sure it's GRCH38 and not a different build like GRCH37, chromosome 20, and it ends with SAM, which is what the output is. So an alternative to this is to go ahead and specify the FASTQ files directly. The form of it is given here, for example, -1 and then some FASTQ files, -2 for the reverse reads, for paired end reads. You can do that, but the beauty of this program is you don't need to. Once you have that accession, you can draw from it right away. Great. So it's still running, and it takes a few minutes to do the alignment. Some projects, such as aligning a whole genome, might take many hours, and for some other software that's not as fast as HiSat, this could take days. What we have, though, is this on-screen output as soon as it finishes, and to give you a preview, it's saying how many reads it detects, 20 million reads, and all of them are unpaired in this particular case, so it's just single end, and that many of them align 0 times, 3.6% overall alignment rate. That's presumably because we've only aligned to the small chromosome 20. So we've got ourselves a SAM file, and that was a great goal to get to. Let's take a quick look. If you go ahead and type head, it'll give you the first 10 lines. I asked for head-4 just to look at the first 4 lines, and this is the SAM file that comes out. It's a text-based file, so you can just look at it with a utility like head or less. And it has some characters in it, and to help make sense of them, first I'd like to give you the link to the sequence alignment map or SAM format specifications that are online. You can read about those to learn about this in detail. Sometimes these are called SAM files, but other people even call them SAM-BAM, because BAM is merely the binary SAM file, which is a compressed version that's commonly used. Well, there are these different mandatory fields in the SAM output, and they may be a little bit hard to see here, but they're all present. And that I'll give you an example from a textbook I just published showing a piece of a SAM file here, and I've tried to color code it. So the query name is given right at the top, and then there's a flag value of 163, a reference chromosome when it's found, and other kinds of information that's very detailed information about the read itself that's shown here in the middle. The base quality scores we saw with the FASTQ file are still here, but there's more information about mapping and about even matches and mismatches. So then for our particular example, we had 710,000 reads that aligned one time, and there were 33,000 instances where something aligned more than once, and that might be because there was repetitive DNA. If you do this with paired end reads, then you can see both the concordant reads and discordant ones, and that can be interesting to look at too. So we got our SAM file. Really, to make it more useful, we just have a couple more steps to go through. SAM tools view, SAM tools sort, and SAM tools index. I'll mention that as we were going through this, the run finished, so that just took a couple minutes. And so we have an output of a SAM file that we can look at, for example, with less my alignment. Here it is, and that less shows us this started off the run with a whole string of N residues because this was aligning probably to the end of the chromosome. As we go forward a ways, we get into reads that actually have more information in them. And so that's all something that you can interpret if you want to go further into it. But really, let's just keep going forward to the end when it all makes even more sense. So SAM tools view is a command that's going to turn your SAM file into a BAM file. SAM tools sort is going to do the necessary task of taking that BAM and sorting it. And this is the argument here is the input is the BAM file, and I named the next one the same thing, except it ends with .sorted. And this is the format appropriate for SAM tools sort, and then SAM tools index of the sorted BAM. And then we are now going to be ready to put this into the final set of software. I'd like to mention that when we turned our SAM into a BAM, the size of the file went down a lot, from several gigabytes down to just 600 some megabytes. And so that's typical that by changing a SAM into a binary form and compressing it, it gets much smaller and easier to use. What do we do now? We have our BAM file, but how do we find out what happened to gene expression? Let's use Stringtie. This is a program that assembles gene models from spliced alignments that HiSat creates. So what Stringtie does is to create gene models even if it has no prior information about them, or it can take known models and quantify those, assembling additional gene models if needed to account for the data that it acquires. The output is either a GFF or a GTF file. GTF stands for Gene Transfer Format, where GFF is General Feature Format. And each of these has interesting fields of information. You can read more about it at a Broad Institute website that describes GFF. So above, in section 6, we obtained a GFF when we downloaded information from Ensemble and also from NCBI. We had it at both places. It's in our reference folder. So to use Stringtie, let's go over here and we'll go clear and then Stringtie. And as usual, you can see what the arguments are. Stringtie requires an input BAM file, and that's just what we have. It needs a -G, then a guide GFF, and then there's an output format here. And the output is often a GTF. There are many, many additional parameters that you can have a look at. So we're going to go ahead and run it. And so we go into the analysis folder, and we can do that by going quit, cd, ... and then move over to analysis. And then in that folder, we can go ahead and run the command. And here it is, Stringtie, my alignment, so this is the BAM file, -G is the specification of your reference genome file, and that's a GFF. The greater than sign is where we're going to send the output, and that's to a file that ends with stringtie.gff. So when we do this, I'll go ahead and start. We go ahead and capture this, and then paste. And I'm going to just give it a slightly different name by adding an extra underscore, and now it's running. So as that runs, let's take a look at what the output will be. There are about 4,700 rows. You can look at it as a text file with a utility such as less. If you do a word count, there are about 4,700 rows in this file. And if you do a head -3, it has a couple hash marks indicating initial lines that have information. For example, a repeat of the command that you ran. and the version of Stringtie. And then it goes into this where it says Stringtie transcript, and it gives, for example, a gene identifier and a transcript identifier particular to the Stringtie analysis. It also gives you a reference ID of a transcript. Here it happens to be with an ensemble accession number, a ref gene ID, and then a gene name, which is actually an official gene symbol, such as ZCCHC3. And then it gives finally information about the coverage and FPKM. So let's take a look at what this output involves. First, it's assembling transcripts, and so note the gene ID and transcript ID fields. Second, it includes already known genes, and that's a nice thing. Third, it tells you how much of each transcript was present in your samples. We've only looked at one sample, but of course you could do this for multiple samples. It reports well-known FPKM values. That stands for fragments per kilobase of transcript per million fragments. The idea of this FPKM concept is we have fragments, which are typically read pairs, that are measured and they correspond to a particular transcript. These need to be normalized based on the total number of fragments that were sequenced. Indeed, there are typically millions of fragments, and this normalization lets us know not an absolute number that doesn't have particular meaning, but a relative number of how much each transcript was expressed. Also, we need to normalize by the length of the transcript. Imagine that you have one transcript of length, let's say 5 times bigger than another one. Gene A is 5 times bigger than B. Well, it might very well have 5 times more reads, but that shouldn't imply that it's 5 times more highly expressed. So that transcript actually is normalized by its length, and that is why FPKM is a really popular measure, although there are other metrics to describe how expression is quantitated. And the GFF file also marks coverage, and so we'll see this next. What we can do is go ahead and view the file in IGV, the Integrative Genomics Viewer. This is a really nice program you can get for free, although it requires registration, from the Broad Institute. Download the software. It's a Java-based program. When I launch it, one way that I do it is to go to Finder, Applications, IGV, and then open up the jar file on my Mac. You can also open it up on a PC if you'd like. And the input can be many different file types, including GFF, GTF, BAM files, VCF files. And so what we do is we set the genome under genomes here to HG38, that's our reference genome. Under the file menu just to its left, we load a BAM file and the GFF we just generated, and here are their names. By the way, as we've been speaking, this went ahead and finished the Stringtie run, so we could do less my alignment 1, chromosome 20, Stringtie.gff, and we can see that it produces information about exons, transcripts, and it gives Stringtie here .3, .4, different models that it's creating as it goes along the length of chromosome 20, and it gives data on coverage and the FPKM value I just mentioned to tell you whether transcripts are expressed at a high level or a low level. I can hit the space bar here to browse through this file and see that many of these transcripts do correspond to known exonic regions. So what I'd like to suggest we do with IGV is navigate to a particular place, chromosome 20, from 3,030,000 to 3,050,000 in IGV. And let's just take a look at what IGV is showing you from top to bottom. We loaded the file in the genomes at the top left. We set the particular genome build here. We're on chromosome 20 because that's what our data has been aligned to. And I specified a particular region. In this box, you can also enter a gene symbol if you'd like for example, GATA5 on chromosome 20, or any gene you're interested in. Next, we see an ideogram of chromosome 20. The telomeres at either end, the centromere is pinched in, and here there's a red bar indicating the position on 20p13. That's the short arm of chromosome 20, where we're currently looking as we zoom in to a 19 kilobase region. In that region, we have tracks, and one track is from the BAM file, and it actually has 2 different parts. The upper part has what looks like a gray mountain range, and this is a really nice way to visualize the coverage. So you can see that these mountain range peaks are here, and in a moment I'll show you the gene track lower down shows gene models that are annotated already, and that includes known exons. And you can see that these gray peaks correspond to the exons of a number of different genes. So the next section here is the BAM file itself. And on this, you can see that there are sort of peach-colored and light blue-colored boxes. Those are paired end reads. And you can see that they have a line connecting them. Sometimes they're present quite close to each other, as we might expect, since they come from one contiguous fragment. But sometimes, because of the nature of exons and how introns intervene and are spliced away, we may be able to see exonic sequences in which either end of a pair is very far away on adjacent exons. And indeed, this is the kind of information that Stringtie uses to build up a model. And so here's an example to the right, where the gene is MRPS26, and that you can see 3 examples of lines going across that connect the exons. And then as we go further down, we see the genes. You can load additional annotation tracks that are pre-configured for IGV. For example, if you wanted to, you could open up repeat masker tracks to see repetitive DNA elements across a chromosome. And then I loaded the GFF down below that. We can just take a look at the IGV live. And so this is what it looks like. As you use IGV, it's really versatile. It's sometimes called the Google Maps of the genome because it's a great way to find your way around. You can simply slide across to look at different places. And then as you, for example, click here, you can show the reads in a different form that's squished so you get a better overview. Or you can go back to where it was, which was expanded. You can color these reads in different ways. We'll color the alignments, for example, by sample, although in this case they're all from the same sample. And then sometimes IGV can show you variants in which there is a reference read and that differs in the actual sequence read that's given here. So there's lots of information you can get from it. And then here with the other panels, you can also click on the side to see different options. And so here we'll make this expanded. You can see different gene models as you go across. And then here we are in an intergenic region, and you can see occasional reads that occur, including also in intronic regions, you can see some reads. And so you might build up support for there actually being a different model than what is currently annotated. And the ENCODE project has described the pervasive nature of transcription across the genome, so you can get some evidence for that as well. If you zoom out, which is in the top right, at some point, fairly soon, the view is now 10 kilobases. If we take it to 40 kilobases, we can no longer see those alignments, although we can still see Stringtie models when they're present. And so here's another model that exists, for example. Why might we not be seeing expression along certain genes at all? Well, this happens to be the context-dependent nature of RNA and RNA expression. It may be that the cell line that's being studied is a lymphoblastoid cell line derived from a blood draw from this individual, and the accession for this one was GM12878. Maybe oxytocin and vasopressin, which are these genes here, are expressed in neurons but not in blood cells, and so we simply don't see evidence for reads along these genes. And so that's just, again, an additional nature of this. So that was IGV, and this is another example of zooming in further. I'd like to mention that you can also go to ClinVar and use a viewer there. If you visit that viewer, you can upload your GFF file as well. And so if I go to Variation View ClinVar, you can go to your data and hit the plus sign here, and then you can add files. And as it's mentioned here, the supported file types include BED, GFF3, GTF, GVF, VCF, and so there's quite a number of different kinds of file, and that when you upload them, you simply would browse to wherever you would like on your computer to upload the files. You then can go to the tracks menu, which is this gear-shaped icon, configure the tracks, and specify what your uploaded data is, and then you can go ahead and configure that and load it. And so this is something else that you may wish to explore. This brings us to the end of our workshop on this section on the topic of RNA-seq. I'd like to close by saying that we've taken a look at next-generation sequence data to find expressed RNA transcripts. And in terms of what's next, this was just a beginning. You might be interested in exploring many different ways. One direction you could consider going is to learn about the R language. It's incredibly popular in the biostatistics community and the bioinformatics community as well. It includes a variety of packages for analyzing RNA-seq data, but also many other genomics topics, not to mention proteomics. So if you want to install R, it's freely available from rproject.org. You can use it on PC or Mac or Linux. I highly recommend RStudio, which is a great environment to be working in R, from rstudio.com. And then once you've loaded your R, you can run it through a really nice interactive GUI. Then that is a graphical user interface. You can try Swirl, which is a wonderful and brief introduction to R, so you can learn a little bit about it. And then go explore Bioconductor, a website that has over 1,000 R packages. Each of them is really well described. And then use R, for example, the ball gown package will let you take a look at differential expression of your RNA-seq data and go explore in even further details. Okay, and that's the end of RNA-seq for now.